Welcome to another microbiology lecture. In this lecture, we will be discussing about the influenza virus. What is influenza virus? Influenza, commonly known as the flu, is an infection of the nose, throat, and the lungs. It is caused by the influenza viruses, which belongs to the family Orthomyxoviridae. These viruses affects the birds and the mammals. Now, the types of the influenza virus. The first type, influenza, is the most serious type of the influenza viruses. It can cause flu pandemics and epidemics. The second type is the influenza B, which can cause seasonal outbreaks, but pandemics generally do not occur with influenza B viruses. Humans are the natural host of the influenza B. The third type of the influenza viruses, the influenza C virus infections, generally cause mild illness and are not thought to cause human epidemics. The fourth, influenza D viruses, primarily affect cattle with transfer to other animals but are not known to cause illness in humans. Influenza can be caused by transfer of the virus from an already infected person or animal. It is transferred through different mechanisms like airborne droplets, contaminated surfaces, or close contact. Inhaling droplets from a person with the flu, typically within a 6 foot radius, can cause influenza. Or touching surfaces contaminated with the virus and then touching the mouth, nose, or eyes can also cause influenza, or direct physical contact with an infected person. Structure of influenza viruses. Influenza viruses are primarily spherical but can also appear as elongated or filamentous particles exhibiting a variable shape, also termed as pleomorphic. They are covered with a layer of spikes. There are two types of spikes present, the rod-shaped hemagglutinin and mushroom-shaped neuraminidase. There are two types of spikes, each made of different proteins. The first one is hemagglutinin. Hemagglutinin enables the virus to attach and enter a host cell, starting the infection. The second one is neuraminidase. It helps new virus particles exit the host cell so they can infect other cells. These proteins are visible to the immune system. The genetic material of influenza viruses comprises of single-stranded RNA which carries the genetic material of the virus. The RNA nucleoprotein is covered by lipid envelope in which the hemagglutinin and neuraminidase spikes are embedded. As we have already discussed that influenza is caused only when the viruses are transferred from already infected individual to the susceptible individual. This transfer of virus can occur by airborne in the form of droplets within a 5 micrometer diameter or a direct contact and indirect contact. There are several methods to diagnose influenza. The first one is rapid influenza diagnostic test RIDTs. RIDTs detects viral antigens. It is quick but less sensitive. The second and the best method is reverse transcription polymerase chain reaction RT-PCR. RT-PCR is highly sensitive and specific, which detects viral RNA. There are two other methods, viral culture or serology, which can also diagnose influenza. Common symptoms of influenza include sudden fever, chills, body aches, headache, fatigue, sore throat, dry cough, and runny nose. Whenever the flu season arrives, it is important to be aware of the symptoms of the influenza. For the treatment of influenza, the patients are given fluids and rest. Analgesics and antivirals are also given to give relief from the symptoms. Drugs like antivirals are also given. These drugs are chemically related antiviral medications known as neuraminidase inhibitors that block the viral neuraminidase enzyme and have activity against both influenza A and B viruses. It is better to follow precautionary measures than treating influenza.